Okay, now they've come up.
Okay.
You are good to go.
Okay. So let me first give an introduction to what is eukaryote. What is the process of eukaryotic genome annotation? So when we're talking about genome annotation, then there's generally two steps. The structure annotation, which is identifying where the start and stop of genes and exons are on the genome, and then the functional annotation, where we annotate the actual function of the genes. I'm focusing on the first one, the structural annotation, which you do when you have a new genome. And this is by nature an iterative process. So the first thing that you need to do is that you mask out any repetitive elements that are going to cause errors in searching where your structures are on the genome, and then you align evidence and use this aligned evidence, which comes in the form of proteins from a related species or transcripts from the same species. And you use this aligned evidence to train gene models, which you can use to predict where genes are, and then look at the combination of all the evidence to assess the gene predictions and refine the models and perhaps rerun the gene prediction. So here, this picture is actually from the SASBi Eukaryotic Genome Annotation pipeline that shows how they do the various steps. I was involved a few years ago in the annotation of the Asian seabass genome. So this Litis calcarifer, it's a teleost fish. It's of economic importance. They call it the barramundi in Australia, and they like buying it. It's also of scientific importance because it is born male and becomes female later in life. So studying the sex change is quite interesting. Yeah, so but it's almost 700 megabase genome. It's diploid. It's got 12 chromosomes and a varying number of accessory chromosomes. So the initial annotation was done using a non-automated pipeline where we had to try and write down every step that we did as we ran the commands. I have a long notebook for this. And it was subsequently re-annotated by SASBi. So automated genome annotation pipelines do exist, like Maker2, Breaker, Eugene, etc. And they combine multiple tools to produce the structured annotation of eukaryotic genomes, but they're kind of black boxes. So typically these are command line utilities configured via text files, which are quite difficult for people who are not familiar with the command line to use. And they support a fixed set of tools and algorithms. And if you want to change that, then you need to go and change the code of the pipeline. Finally, they have no support for tracking data provenance. This proved to be quite challenging for us when we were working on the Litis calcarifer genome, in that, you know, when the reviewer asks you to go back a year in time and clarify how you came up with a particular result, but you were running everything on the command line and you don't remember exactly which parameters you used, or maybe you forgot to write down one step that you thought was insignificant at the time. So yeah, the Galaxy project, we've heard about it a bit earlier. It's a web-based framework for accessible and reproducible bioinformatics. Galaxy can run individual tools, or it can run workflows, which are built out of these tool or algorithm building blocks. And then when you've got a workflow, then the logic is visible and can be edited in this workflow editor without knowing programming or the Linux command line. So the data in Galaxy is stored in these things called histories, which is a user interface that stores the provenance of each piece of data. As you run a tool, then you can always go back and rerun the tool or examine what was used as input and as parameters for the tool at some point in the past. So there has been some work with the G-ONRAM project in Galaxy Genome Annotation, collaboration on building tools for eukaryotic genome annotation in Galaxy, but it's still at a kind of basic stage. So I decided to take this platform and take it further. So this is what Galaxy looks like. On the left is a selection of tools. On the right is the data, which you are in the so-called history. And in the middle, there's some information about either what's going on when you run Galaxy, a tool in Galaxy, or you can view your data here or see the input form for a tool. Here, I've just pasted a picture of a very simple Galaxy workflow. So any of these tools here on the left, you can drag onto this canvas and connect them together with these little noodly lines. And what this little workflow was for was that the Spawn tool, that is an evidence mapping tool, its output was a little bit dirty, so I was just running it through some text reformatting and some sorting of the resulting GFF so that I could use it as input to JBrowse, which is a genome browser that you can run within Galaxy. You can run JBrowse outside of Galaxy as well, but just one of the things you can run within Galaxy. So you can make much more complex pipelines than this, but I just wanted to illustrate a simple one on the screen. So there are some challenges, unfortunately, in the process of boarding on genome annotation tools. Firstly, I'm trying to work in an open science way, but the licenses for tools and data are sometimes not open. One shocker for me in the last couple of years is that the most commonly used repeats database, RepBase, is now $1,700 US per researcher per year to use. Then when I looked at a ab initio repeat annotation tool called RED that had some very interesting publications in the literature, I found that the code is out there, but there's no published license. So it's not at all clear under which circumstances you can reuse it and share it with others. And a lot of the classic tools in this field, like the GeneMark ES, Gene Predictor, have this kind of free for academics license, which means that if you want to build a set of tools for redistribution to other people, well, then you don't know whether those other people are academics, and therefore you can't include non-open source tools like this. Unfortunately, also the software quality of bioinformatics tools is sometimes poor, causing them to crash. And this might be because eukaryotic de novo annotation of eukaryotic genomes is a somewhat obscure field with a small community of scientists boarding on the same software for years without need to actually, have other people look at the software and perhaps improve it. In fact, one of the tools that I ended up using, as far as I can tell, been under continuous development since the 90s at least. Finally, Galaxy is a nice platform for boarding on, but if I need to add a new data type to Galaxy, then it has to get added to the main Galaxy code base. So besides the technical challenge of that, if you want to use it in a normal Galaxy server without all sorts of patches, you have to wait for the next release cycle, which takes about 6 months. Okay, so what's working thus far? I have also been working on making the setup of a server for genome annotation reproducible. So I have a way to automate the setup of my masters-oriented Galaxy server. I've identified repeats using the window masker tool, which is an ab initio repeat identification tool that NCBI actually uses internally, because identifying repeats from databases like RedBase, besides the cost, is not that reliable for genomes where the repetitive structure is not very well known. And I've written a suite of tools for working with the SASBi taxonomy database, specifically because I'm interested in filtering the RefSeq protein database by the taxon. So if you want to find all of the proteins which are associated with ray finned fish, then that is the kind of thing that my tool suite can do. And at the moment, I've been focusing on tools and workflows for working with evidence. So Spawn, which I've mentioned previously, and a tool for selecting the best model for optimizing Spawn parameters, and some RNA mapping workflows with HiSat2 and string type. So here's an example which illustrates why it's important to be able to rerun eukaryotic genome annotation with different parameters. I was looking into the evidence around one particular protein, the CYP20A1 protein, And we can see here this JBrowse genome browser is actually running within Galaxy. We can see some predictions from Augustus, which seems to have merged some genes together. From Spawn, which this black gene over here is from a related species, the tongue sole. It's relatively closely related to the Asian seabass. And then two different sets of prediction from RNA-seq evidence. So here at the top, we've got the NCBI annotation, which says this is all one gene here. And down here, we can see that some of the prediction is split in two. So I went to go see why that's happening. And that's because if you look at these RNA-seq reads at the bottom, then they get fragmented by the fact that there is a repetitive element masked out here in the genome. So rerunning it the second time without the masked version of the genome at this part showed that there's actually a link between these two pieces of the gene, which allowed me to see the complete gene structure like this. So being able to rerun genome annotation with different parameters is definitely useful when exploring structural annotation. So what next? I'm working on some faster tools for evidence mapping, incorporating some of the de novo gene prediction tools like SNAP and Augustus, the ones that are open source, and building the final pieces to consolidate all the evidence into actual gene predictions, and then finally re-annotate the complete lattice calcarifer genome within Galaxy. And yeah, thank you to everybody at SASBi, especially my supervisor, Professor Alan Christoffels.
Great. Thank you very much, Peter. Lots of virtual applause. Really enjoyed the talk. What a lot of work you've done. It's really an incredible amount to put into one presentation. And there's already lots of questions in the chat, so I'll go straight to the ones from our participants. First question, to your knowledge, can this be used for microbiome data analysis if it hasn't already been used for microbiome analysis? So I think Galaxy.
Galaxy has been used for microbiome data analysis, and any other Galaxy questions, please hit me up maybe during the social later. But I'm focusing on the case where you have a novel single genome from a eukaryotic species.
Okay, so focusing on new genomes in your case.
New genomes, because it's becoming quite easy to do a new genome. Well, not 100% easy, but relatively easier to do a new genome. But then the question is, well, where are the genes?
Yeah, of course. And following that, the mass question, what do they do?
Yeah, the functional annotation, yes.
Yeah. Another question, where do you draw the line between annotating a plant versus an animal genome in terms of annotation? Do you have any...
Yeah, sorry, carry
on. Do you have any recommendations based on the repeat content, size of the genome, or any other genome characteristics to choose an adequate genome annotation tool?
Yeah, so firstly, the repeat, I think there's a whole lot of work that needs to be done on better tools for annotating repeats. Broadly, there's two concerns here. The one is actually understanding what the repeats are, and secondly, just identifying repetitive regions, okay? So the existing repeat databases don't work very well when you start stepping outside of the well-known mammalian genomes. I don't have specific recommendations on annotating repeats in plants, though. One of the concerns that I've got about genomes is what to do when they get much larger, which is why I'm working on tools for making the evidence mapping stage faster. And in terms of recommendations for pipelines, there are some. For instance, Eugene has specifically been designed from a group that is mostly working on plants. But I'm hoping that within the next 6 months, I can give you a new recommendation for a pipeline for working on your genome annotation.
Different plants, of course. And their follow-up was, following that, if you have this pipeline available, is memory going to be a problem? So what about memory usage if you were annotating a plant genome of 600 megabases?
Well, the thing is that what you tend to do is break everything into chunks. So you look at proteins you break up the collection of proteins from your related genome into chunks, and also the context that you're annotating. The memory problem tends to be much more of a problem when you're doing the actual assembly. So I know that some of my colleagues have been working on robots, and they did some of the stages on a machine at the CHBC with like a terabyte of RAM, because doing your kind of the brain graph assembly of large genomes is very, very memory hungry. But when you're doing the annotation, memory is less of a concern.
I see. So you have to pick your strategy then, depending on which jobs to run where.
Yeah, if the assembly versus annotation. And if we're lucky, CHPC will host a Galaxy server at some point, and then we will have access to all of that power.
Amazing. Peter, we're grilling you with questions. They keep popping up into the chat. It is actually time for our break. How do you feel? Can you take some more?
Just a couple more.
All right.
We have a social time later as well, I think.
All righty. So 2 more questions. You mentioned that you've developed your own tools. Have you shared them, or are you developing them for your own use? If you did share them, do you have a GitHub? And once you have them on a GitHub, is there a way to pull that into your own Galaxy structure?
Yeah, so everything that I do goes straight onto GitHub. Everything from the setting up the actual Galaxy server, all of the tools are developed in collaboration with the Galaxy so-called IUC, their tool development community. So everything that I'm doing is open source, and as soon as I have a tool available, then I commit it to the GitHub repository of the IUC, and you can install it on your own Galaxy server. Yeah, it's not all consolidated in one place yet. That's the point of my master's, I guess. But I'm committed to making everything available as soon as it's done.
Awesome. And then one final question. Has anybody, maybe not just for you, Peter, but has anybody used Galaxy on organisms other than plant or animal species, maybe some such as fungi? What are the accuracy of annotations in terms of these non-model organisms? Or would you preferably use software such as Maker?
So Maker is one of the tools that I'm actually using. So Maker uses different pipelines internally, but for instance, for mapping protein and transcript evidence, it uses BLAST followed by exonerate. One of my critiques of Maker is that BLAST and exonerate might not be the tool collection that you want to use. So then if you want to do things differently, it's very difficult to change Maker. But where I'm going to use Maker as a component is Maker has very nice tools for amalgamating all the different evidence streams into gene prediction. So what you can do is use other pipelines for producing evidence predictions, and then use Maker just as the step that basically kind of puts everything together. And in terms of fungi and Galaxy, I know somebody from the University of Jos in Nigeria who's actually annotating the genome of a Candida species. They're using the usegalaxy.eu server because they don't have local compute resources. So definitely, Galaxy is a platform. It does whatever you basically can make it do. Outside of my master's, I've used it quite extensively for work on bacteria. So in fact, we've just recently released a tutorial on Mycobacterium tuberculosis variant annotation in Galaxy, which you can find on training.galaxyproject.org. So yeah, it's not restricted to one domain of life or one domain of bioinformatics.
Amazing. Peter, you're a legend. Thank you for answering all these questions. I'm sure you'll have many more later on in our social settings. And right now, we're on a break. So thank you, participants. Thank you to all our previous speakers. Really enjoyed all your talks. We look forward to the next set after our break. And as you can see on the screen, there is a little bit of a poll going on, just asking you about whether you have attended a SASBi student symposium before. Please go ahead and fill in that poll. It'll be cool for us to see what we're doing in terms of a virtual symposium versus our in-person symposia. It's just good to get feedback from you guys, maybe for the future, for a post-COVID world, to see what we can do in terms of sharing our science. All right. Thank you again, Peter, for being such a champion.